Hello, my name is Joshua Burrows. I'm a fourth-year PhD student here in the UCSD Systems Biology Research Group, and I'm excited to talk to you a little bit today about my first paper. This paper is titled "Structure of the Enterobacter Pangenome is Revealed Using Machine Learning." It's now available in Microbiology Spectrum, and I'm excited today to talk to you a little about some of the results within this paper and its importance as a study of the understudied nosocomial pathogen Enterobacter. So with that, let's jump into some of the results. First of all, I'd like to talk a little bit about some of the motivation underlying this study. One of the vital first overall motivations is Enterobacter status as an escapee pathogen. The escapee pathogens are very common nosocomially infectious pathogens or hospital infectious pathogens that have high mortality rates and high rates of resistance to antibiotics. This resistance to antibiotics or antimicrobial resistance, AMR capabilities of these organisms make them very important for further study, as the increased spread of antibiotic resistance makes the antibiotics which we use to treat these infections less effective over time and necessitates further understanding of how to combat these mechanisms and understand when, where, and why these mechanisms are present across these different species genuses of interest. One of the second motivations of this study is the understudied nature of Enterobacter as compared to many of the other nosocomially infectious pathogens that are more understood, such as the other members of the escapee group, as well as E. coli, a pathogen which is related to Enterobacter somewhat closely. They're in the same family of bacteria. However, there's a very large gap in the understanding that we possess between some of the most studied species of bacteria and genuses such as Enterobacter, which are largely understudied, although there has been a lot of great work in the field of study of Enterobacter. So much more room to better and further understand this vital pathogen. Getting into the results of the study, we first need to talk about the collection of the strains for our pangenome. There are 777 high-quality complete sequences will be collected from a variety of databases, including the NCBI database and VVBRC, a large database which collects genomes for pathogens such as bacteria and viruses. We quality controlled them and selected them for the construction of our pangenome. These genomes come from a variety of different sources, many of which are clinical, which is expected for our nosocomial pathogen Enterobacter. However, they come from a variety of different sources as well, such as wastewater and other environmental samples, but also from plants and animals. The collection of Enterobacter strains here spawn a variety of different species and a number of subspecies of the most common pathogen within the genus, that being Enterobacter or mache. After gathering this large collection of strains, we need to construct the pangenome. Construction of the pangenome allows us to go beyond looking at the genome of one strain at a time to looking at the genomes of dozens, or in this case, hundreds of strains at a time, in order to gain more robust and thorough information about the underlying genetics, not of a single strain of Enterobacter, but of Enterobacter as a whole. In construction of this pangenome, we see which genes are present across which strains of Enterobacter and how they're shared across the pangenome. And this results in three different categories of genes within our pangenome. There's the core, accessory, and rare genomes. The core genome represents genes that are found across all or nearly all strains in the pangenome, often representative of key cellular processes such as respiration, key metabolic processes such as glycolysis, as well as some unique genes such as the beta-lactamase, which is present throughout virtually all of Enterobacter. This is an antibiotic resistance gene, which is used to resist penicillins in bacteria. It represents one of the many ways in which these Enterobacter species are able to combat antibiotics. However, the core genome does not do a great job in differentiating between different strains, species, or subspecies of Enterobacter because it's shared throughout all or virtually all of Enterobacter. This leads us to the accessory and rare genome. The rare genome is found in very small numbers of strains throughout our pangenome and can provide a variety of important traits of interest, but is not very useful in determining groups of Enterobacter, as it is so rare that it is often not able to distinguish more than one strain at a time, or with a gene only being present in one or two strains in most cases for rare genes. The accessory genome is where the majority of the study lies in this paper. The accessory genome represents genes that are found in anywhere between 97 and 7% of the strains, and can there be representative of genes which are common to large populations of Enterobacter and also differentiate between these large populations of Enterobacter. However, we once again run into the problem of, with such a large number of strains, how are we going to determine how these genes are differentiating Enterobacter and leading to the different traits that these species and subspecies have? Well, that's where our analysis technique comes in, non-negative matrix factorization, or NMF. NMF takes our large matrix of genes and strains, where the matrix is representing which genes are found across which strains. There are thousands of genes in the accessory genome and hundreds of strains. It's difficult to interpret this matrix as is. NMF allows us to take this complicated large matrix and decompose it into two more interpretable matrices. A matrix of genes by components and components by the affinity of the different strains for each of these components. These components are called phylons, and they represent groups of genes which co-occur across different subgroups of Enterobacter throughout our collection. These phylons can be broken into two major categories, the lineage-associated phylons and the mobile phylons. The lineage-associated phylons are representative of vertically inherited sets of genes across our pangenome. These lineage-associated phylons are often directly associated with different species or subspecies of Enterobacter, giving us insights into the genes which define these different groups of Enterobacter in a very clear and formulaic sense, allowing us to identify as a group what are the traits that are most commonly shared throughout a certain species, subspecies, or in some cases, even smaller subsets of subspecies in our pangenome. Some of the important traits which can be differentiated here are AMR genes, which are common to specific species or subspecies of Enterobacter, as well as certain biosynthetic gene clusters, clusters of genes which allow synthesis of important compounds relevant to certain species or subspecies in the niches in which they live. The redox cofactor that is found throughout a variety of different Hormache species assists in their metabolic capabilities, as well as throughout a variety of different studies has been seen to improve their potential as a pathogenic organism. This operon is found across Hormache species as a species, but not any of the other species of Enterobacter. An unexpected result on this front was finding a homoserine lactone operon or biosynthetic gene cluster in Roentgen-Campi and his Burrows strains. This has been reported before in individual strains, but our study allowed us to confirm that this biosynthetic gene cluster was found throughout virtually all strains of Roentgen-Campi and his Burrows, displaying that this compound, homoserine lactone, which is used in quorum sensing or communication between bacteria in different communities, is important to their survival or adaptation to the niches in which they live, differentiating them from other species of Enterobacter. The other portion of the phylons that is present are the mobile phylons. These mobile phylons are representative of primarily horizontal gene transfer events. For the most part, in our study here, plasmids. These plasmids contain a variety of very important traits of interest and are not simply associated with one species or subspecies of Enterobacter, but are found throughout strains across the pangenome from a variety of these different species and subspecies. The different traits present in these mobile phylons include antibiotic resistance genes, which are of vital importance for study of this pathogen, but also traits that might be a little bit less expected, such as heavy metal resistance genes that are found throughout clinical and environmental samples for these organisms. This gives us insight as to the importance of these heavy metal resistance operons due to their repeated acquisition through horizontal gene transfer for this organism, necessitating further study of both why this is occurring and giving us insights as to some of the stress challenges that these organisms face and how horizontal gene transfer enables them to acquire genes which make their survival more robust. Some of the major technical issues that we face within the study were adapting some of our NMF techniques from a single species, such as E. coli, to Enterobacter. This required adaptations in determining how many phylons that you're going to use or how many phylons you're going to decompose your matrix into. Advancement of our rank selection techniques and algorithmic approaches to this was a key challenge in this study. But one of the most central challenges to the study was applying this technique to an understudied organism such as Enterobacter. Ensuring consistency with taxonomic naming conventions for Enterobacter, getting up to date on all the current literature for this organism, and ensuring consistency with the, while limited compared to E. coli, but equally important study that has already been done in this organism was a vital part of this study, so that we are advancing the knowledge present for this organism by combining what is already known with some of the unknown parts of information that we found throughout this study. We can clearly see the accessory genome of Enterobacter can be used to define the differences between vitally important populations and subpopulations within the Enterobacter genus, allowing us to see the genes which make up key species and subspecies within Enterobacter, but also to see the importance of horizontal gene transfer in the acquisition of traits of interest for this genus, such as antimicrobial resistance genes on plasmids, but also heavy metal resistance genes on plasmids. The accessory genome can therefore define both vertically inherited sets of genes, lineage-associated phylons, which give us insight into the evolution of strains to their different niches, as well as horizontal gene transfer of these vital genes, which enable more robust survival strategies of Enterobacter across a variety of different niches. Determination of these phylons offers a very robust and clear breakdown of what is known about Enterobacter so far by taking a large collection of genomes and compressing that information to make it more interpretable, clear, and relevant to researchers who want to spend more time understanding Enterobacter, both developing some of the fundamental science principles behind the study of Enterobacter, as well as key findings towards understanding this organism clinically, such as AMR resistance, heavy metal resistance, availability of genes to these organisms, etc. With that, I'd like to thank you so much for listening, and I encourage you to go check out this paper in Microbiology Spectrum.